Namaskaram students. In the last class, we discussed about mutation and its types. In detail, we saw the gene mutation or point mutation. Today we are going to discuss about chromosomal mutation. What is chromosomal mutation? And altering the chromosome, either its structure or its number. As a result, this type of mutation is called as chromosomal mutation. It is also called the name as chromosomal aberration. Whereas in gene mutation, such mutation occurs within a gene. But in chromosomal mutation, the mutation can seen in not only single gene, we can seen in many genes. How we know whether the mutation is occurred or not? Gene mutation, it is not possible to analyze. Whereas in chromosomal mutation, it can be detected by microscopic examination, genetic analysis, or both the one. So this chromosomal mutations is divided into two types. They are based upon the number as well as based upon the structure. So we have to call chromosome number and chromosomal structure in mutation. Let's see in detail about what is chromosomal mutation in number. This is also called the name as ploidy. So every organism can possess the fixed number of chromosomes. For example, human being having 46 chromosomes, onion having 8 chromosomes, rice having 12 chromosomes. So every species they have the fixed number of chromosome, but this may be vary in different species. In plants and animals, they have the identical number of chromosome, but they don't have the similar characters. Though human being, we are all have 46 chromosome, whether we are cloned, no. Everyone have the different physical, morphological and behavioral expression to be different. So this is based upon the expression of the chromosome which is seen in different organism. It is not be same, it should be different for the different species. And these are expressed based upon the chromosomal characters. So sometimes this chromosome number which is seen in somatic cells, they can be changed due to either addition or deletion of the chromosomes or the entire set of chromosomes. This is called as numerical chromosome aberration or ploidy. Again I am recalling if any changes in the number of chromosome, we have to call the type is called as numerical chromosomal aberration or ploidy. Sometimes this chromosomes based upon the either addition or deletion, again it is classified into two types. They are aneuploidy and euploidy. In aneuploidy, numerical aberration seen in individual chromosomes but within a diploid set. Just keep it in the mind. In aneuploidy, individual chromosome changes occurs within a diploid set. Whereas in euploidy, the chromosomal aberration occurs in entire sets of chromosome. Whole sets of chromosome is going to be changed. Whereas here, individual chromosomes alone, they are going to change. This is the difference between aneuploidy and euploidy. Aneuploidy is divided into two types. They are hyperploidy and hypoploidy. Hyper means addition, hypo means deletion. In hyperploidy, addition of one or more chromosome from the diploid set. Whereas in hypoploidy, loss of one or more chromosome from the diploid set. Next in detail we are going to see about the aneuploidy. It is divided into two types. They are hyperploidy and hypoploidy. Aneuploidy means it is a condition in which diploid set of chromosome is altered either by addition or deletion. It is also called the name as heteroploids. Now we are going to see about hyperploidy alone. Hyperploidy, again they are subdivided into 5 types. Let's see one by one. The first one is called as trisomy. Before that, what is hyperploidy? Addition of one or more chromosome from the diploid set. I already said hyper means addition. So addition of chromosome is called as hyperploidy. This is again divided into various types. Let's see the first one. Trisomy. What's the meaning of tri? Tri means 3. So already we have two set of chromosome diploid. In addition to that, one chromosome is going to be added. So they are called, the name is trisomy. Addition of single chromosome to the diploid set. And this was first reported by Xll in the year 1910. It is also noticed in the Datura stromanium, Nicotiana, Pisum and Enothera. And second type is called as a double trisomy. Just to see the number 2n plus 1 plus 1. If they have 2n plus single 1 means that is trisomy. Again they are given added the one more gene. So they are called, the name is double trisomy. This is nothing but addition of two individual chromosomes from the triploid set. Sorry girls. Addition of chromosomes, two chromosomes from the diploid set. This is called as a double trisomy. And next we are move on to the tetrasomy. Tetra means 4. Already two set of chromosome. In addition to that, again 2 is added. That is called as tetrasomy. Addition of two individual pairs in along with the diploid set. This is called as tetrasomy. We can see such type of tetrasomy in wheat. Next one is a double tetrasomy. Again, 2 times the tetrasomy is going to be added. That is called as a double tetrasomy. 2n plus 2 plus 2. This is also they are noticed in wheat plants. And last type is called as a pentasomy. Penta means 5. That is addition of 3 individual chromosome from the normal diploid set is called as pentasomy. Again I am saying trisomy means 2n plus 1. Single chromosome they are going added. Tetrasomy means 2 chromosome they are going to add. And pentasomy means 2n plus 3. So that's all about the hyperploidy. And next we are move on to the second type in aneuploidy is hypoploidy. Hypo means loss in number or reduce in the number of chromosome is called as hypoploidy. So loss of one or more chromosome from the diploid set is called as hypoploidy. Again it is divided into various types. Let's see. The first one is called as a monosomy. Mono means single. So from the diploid set, one chromosome is going to be lost. So they are called, the name is monosomy. Loss of single chromosome from the diploid set. This is the example for monosomy. And what is double monosomy? Here, loss of single chromosome which is present in the 2 times. That is called as a double monosomy. 2n minus 1, again 1 minus 1. That is seen in maize plant. And next we have to move on to the triple monosomy. That means 2n from the diploid set, 1 chromosome is lost. Again 1 chromosome is lost. Again 1 chromosome is lost. So we have to call the type is triple monosomy. 2n minus 1, minus 1 and minus 1. And last is called as a nullisomy. Nulli means nothing. 2n minus 2. 2 set of chromosome here lost. So they are called, the name is nullisomy. Loss of pair of chromosome from the diploid set. Diploid set already they have the 2 set of chromosome. That is going to be lost. So there is no net gain is 0. So they are given the name is nullisomy. And double nullisomy means loss of 2 pair of homologous chromosome from the diploid set. 1 pair means we have to call the name as nullisomy. Loss of 2 pair means we have to call the name as double nullisomy. And such type of nullisomy we are selfing means selfing the monosomy plants produce the nullisomy that leads to the death of an organism. So we have to call the name as lethal. So as a result of double nullisomy, the plants going to the death conditions called the name as lethal in nature. And again we are going to recall the types of aneuploidy. Just observe the pictures. The first above the pictures, a organism having the 4 set of chromosomes. They are diploid in nature. Whereas in aneuploidy, the first type is called as a nullisomy. Nulli means the 4th chromosome. 2 chromosomes they are lost. 2n minus 2. The next type is called as a monosomy. Here 1 chromosome is lost is called as a 2n minus 1. Double nullisomy means again double monosomy. Here 1 chromosome which is lost in the 4th chromosome number, and again 1 chromosome number 1 gene is lost in the 3rd chromosome. So they are called, the name is double monosomy. So these 3, nullisomy, monosomy, double monosomy are the example for hypoploidy. And next we are more the example for hyperploidy, trisomic. Let's see the normal chromosome. Fourth chromosome, they have the two diploid sets are present, whereas in trisomic, in addition to the two, again one gene is added. So we have to call the name as 2n plus 1. Tetrasomy, 2 gene is added, 2n plus 2. Double tetrasomy, here 4 chromosomes, which is seen in the chromosome number 4. Again, addition of 2 chromosome in the chromosome number 3. So as a result, 2n plus 2 plus 2. So these three are the example for hyperploidy. So that's all about the aneuploidy. So aneuploidy means changes in the number of individual chromosome, but not in the complete set. Just to keep it in the mind, individual chromosome alone, they are going to be changes in the number. So this can be tolerated in plants, whereas in animals, it's due to the death. But in the higher organism like human being, it causes some of the genetical disorders like sickle cell anemia, multiple sclerosis, thalassemia, etc. Let's see in zoology detail. Again, I am recalling types of aneuploidy, hyperploidy having the more than set of chromosome, whereas hypoploidy, they have the less than number of normal chromosome. And next we will move on to the second type of ploidy is called as a euploidy. Euploidy is a condition in which an organism possesses one or more basic set of chromosome, and this is classified into different types. If the organism having single set of chromosome means we have to call the name as hypoploidy. It is divided into 3 types. They are monoploidy, diploidy and polyploidy. If the organism having the somatic cell, half the number of somatic cell we have to call the name as haploidy. 2 set of chromosomes which is seen in the somatic cell means we have to call the name as diploidy. Again I am saying haploidy and diploidy. During the time of reproduction, a gamete cell is going to be reduced. That is called as haploid. So that process is haploidy. During the fertilization, one gene from the maternal and another gene from the paternal, both are fused together to get the diploidy nature. So that process is called as a diploidy. So single set of chromosome is haploidy, double set of chromosome is diploidy. What is monoploidy? So this is different from haploidy. For example, in wheat plant, is a wheat plant, it is an example for hexaploidy. Hexa means 6. So wheat plant having the chromosome number is 72, but its haploid number n is 36. But if it is a 2n means we are saying that 72. Single n means we have to say the number is 36. But in between they are given the 6 H's. 6 represent the hexa and X represent the how many times they are going to be increases. 12 times if it is multiplied with the 6, we can get the number of progeny is, number of chromosomes is 72. So haploid is different from the monoploidy. So just half the number of chromosome is called as haploidy. That chromosome, when it is multiplied with the other, we can get the same chromosome number is called as a hexaploidy. 6 into 12 is equal to 72. So here all monoploidy can be a haploid, but all the haploids cannot be a monoploid. So this condition is an example for the polyploidy nature. In true diploidy nature, both monoploid and haploid have the same number of chromosomes. So again I am saying polyploidy, it is divided into triploidy, tetraploidy, pentaploidy, hexaploidy. Based upon the entire set of chromosomes is going to be changed is called as a polyploidy. So this polyploidy, again they are subdivided into 2 types. They are autopolyploidy and allopolyploidy. Auto means self, allo means different. Here more than 2 haploid set of chromosomes we are derived within the same species. Auto means self. So possession of more than 2 haploid sets of chromosomes within the same genes or same species is called as autopolyploidy. Whereas in allopolyploidy, the same definition, presence of 2 or more basic set of chromosome which is derived from different species. Keep it in mind, whereas in autoploidy it is derived from the same species, whereas in alloploidy it is derived from the 2 different species. Let's see the autopolyploidy first. The definition again I am recalling, the organism which can possess more than 2 haploid set of chromosomes, but it is seen in the same species, and it is divided into 2 types. They are autotriploids and autotetraploids. So tri means 3 set of chromosomes. They can produced artificially by crossing the 2 different types of genes. One is called as autotetraploid and another is the diploid in nature. Autotetraploid means 4. Diploid means 2. So if you are crossing these two, in the F1 generation they are getting the sterile. This leads to the defective formation. But in the F2 we can get the progeny in the higher rate. For example, in cultivated banana, triploids or emerges here. Seedless fruits we can see in watermelon, apple and tomato. And another plant is called as a one weed plant, Cyanodon dactyne. This is also an example for natural autotriploid. Commonly we have to call the name as doob grass. Whereas in autotetraploid, tetra means 4. Having the 4 set of copies of chromosome, doubling the chromosome to be happen here. So if for example, if any organism having the 2 set of 2n chromosome, it with the help of some chemical they are going to double the chromosomes. 2n into 2 is equal to 4n. So this is called as a autotetraploid, having the 4 set of genes inducing the doubling the number of chromosomes. This is called as a autotetraploid. For example, rye, grapes, alfalfa, groundnut, potato and coffee. That's all about the autopolyploidy. And next we are move on to the allopolyploidy. So allopolyploidy means, again already I said the definition, possession of 1 or 2 more basic set of chromosome which is derived from the different species. Keep it in mind that is derived from the different species. So it is possible for the intercrosses among them. So here fertility can be increased by applying some treatment with the help of the chemicals which is derived from one plant. The name of the plant is Colchium autumnale. So from the plant we can get one chemical compound. The name is alkaloid colchicine. If you are applying this colchicine, it can be either doubling the chromosome or tripling the chromosome. Whatever the number we are need, we can be get. So alloploidy is possible only with the help of the closely related species. And let's see the 2 examples which is given here. Alloploidy, the first experiment which is done by a Russian genetist. The name of the person is Karpichenko in the year 1927. What he did is, he crossed the radish plant with the cabbage. The botanical name of radish is Raphanus sativus, cabbage, Brassica oleracea. So both the type of plant having the same set of chromosomes, 18. When they are crossing these 2 chromosomes in the F1 hybrid, he obtained as a sterile. I already said about the colchicine. It's a one type of chemical which is derived from one plant. If you are applying this chemical to the F1 hybrid plant, as a result it can doubling the chromosome. So that F2 to be a fertile in nature. So 2n having 36. How the 36 arrived? So doubling the chromosome. So 2n is equal to 4h. 4h is equal to 36. It is a tetraploid. 4 into doubling the chromosome with the help of the colchicine is called as the example for allopolyploidy. In Brassica, what he thought is, he expected the result root of the radish and the leaves of the cabbage, but unfortunately the vice versa can be result obtained. So he was greatly disappointed. So what he was observed is, he observed the root of cabbage and the leaves of radish. So this experiment is failed in the Russian genetist. So again I am saying in Raphana brassica, both the one having the same set of chromosome. When they are crossed, the F1 should be a sterile, whereas treated with the colchicine it should be a fertile in nature. But this fertile, what he obtained, the result is not be expected result is not be obtained. The name of the new progeny is called as a Raphano brassica. Here the root of cabbage and the leaves of radish can be obtained. So it is an example for the interspecific hybrid having the complete diploid set of chromosome. So this is an example for allotetraploidy. So they are given the name as 4H. So 2n is equal to 19. Here 2n is equal to 36. 18 into 2. So here 2 times they are increases the double the set of chromosomes. So they are given the name as a 4X. And another one example successfully they are produced is called as a first man-made cereal by crossing the wheat and rye plant. So depending upon the ploidy level, it is divided into 3 types. They are tetraploidy, hexaploidy and octoploidy. So the second experiment is the first successful experiment in the allohexaploidy. Tetraploidy. Here they are crossing the wheat plant with the rye plant. So this is the progeny which is the 4 set of chromosomes are arrived. And second type is called the hexaploidy, wheat having the 4 set of chromosome and the rye having the 2 set of chromosome. Again they are doubling the chromosome with the help of colchicine. We can get the triticale is the product. Here they exhibit both the progeny of both the parental trait of wheat as well as the rye plant. In wheat plant, the Marconi wheat variety have the high protein content. Similarly, in the rye plant having the high amino acid content. So in the progeny, in the F1 and F2 hybrid of the triticale having the expression of both the trait, high protein quality as well as a high amino acid lysine. And third type is called as the octoploidy. So here they are crossing the wheat with rye plant. So wheat is the hexaploidy and rye is the diploid. So as a result, we can get the triticale is in the octoploidy condition. So these 3 example based upon the ploidy level. Next, in detail we are going to see about the triticale, how they are arranged. So triticale is the first man-made cereal obtained from cross between the wheat and rye. So the botanical name of wheat is Triticum durum and the rye plant name is Xll Serenale. So triticum having the 28 set of chromosomes and a cereal having the 14 set of chromosomes. So 2n is equal to 4h. Here 4 represent the tetraploidy and 2n is equal to 2h. 2h represent the diploidy. So here tetraploidy and diploidy they are crossed. We have to call the name as autopolyploidy. Sorry, it's a allopolyploidy because they are different plant species. So it is going for the gamete formation. From the 2n, single n is formed is equal to 2h is equal to 14. Whereas in cereal, n is equal to x is equal to 7. Allowing them to be for the F1 hybrid, we can get 2n is equal to 3h is equal to 21. So this is not the triploidy. It is again by treating with the colchicine treatment, the triploidy is turned into the hexaploidy. So in the F1 hybrid, they are getting the triploidy, but the result is just sterile in nature. When we are applied with the same chemical, the name of the plant is colchicine. What I said earlier, it is a poisonous chemical derived from the Colchicum autumnale. It is a highly alkaloid plant extracted from the root as well as the corms of plant. When it is applied in the lower concentration, it can induces the polyploidy in different types of plants. Whereas we are applying the high concentration, the plant is going to be a lethal in nature. So it is a poisonous character. Again I am recalling, if you are crossing the triticale with the help of Marconi wheat and rye plant. Here rye having the high content of amino acid, whereas wheat having the high content of protein. We are going to cross both the one. As a result, we can get both the traits. So tetraploidy is crossed with a diploid. We can get the triploid in nature, but it is a sterile. When it is applied with the colchicine, doubling the number of chromosomes to be happen. So 2n is equal to 6h is equal to 42. So this is the fertile triticale first man-made cereal plant. It is an example for hexaploidy. And these are the product we can get from the triticale plants. And last we are going to see about what are the significance of ploidy. So many ploidies are highly viable than the diploid in natures, and they can adapt to the various environmental factors. And it is a very pleasant appearance in color. It has a large flower and the duration of the flowering is longer when compared to the diploid. So maybe we can use it for the ornamental plants. And it can increase the fresh weight having the large content of water. And this can determine by phenotypic ratio either the loss or gain. So they have the different in nature of the chromosome number. Many angiosperms or alloploids, they play a very important role for the evolution of the plant species. Again, I am recalling the significance of ploidy. It is a highly bigger than the diploids. That is, a vigorous means viability. and mostly we can be useful for the ornamental purposes because of the larger in size and the longer duration, and it has a high water in content, especially we can see in the musa plant. It is also determine the phenotypic ratio, either a gain or loss. It is play a very important role for the evolution of new plant species. Today I am summarizing. We discuss about chromosomal mutation, in which we details saw about the chromosomal aberration in number alone. In the next class we discuss about structural chromosomal aberration. Thank you girls.